CRISPR-Cas9 is one of the most promising technologies in modern genetics. Discovered in 2012, this system of programmable genome editing has endless potential uses in agriculture, medicine, and genetic research. We're going to take a look at some of the human medical applications of CRISPR, along with the ethical issues that may arise. So how does it work? The CRISPR-Cas9 system contains a Cas9 enzyme bound to a guide RNA. Cas9 unwinds and cleaves the DNA. The guide RNA allows for site specificity through its protospacer, a 20 nucleotide long region that's complementary to a target sequence. Any gene of interest we want to edit or knock out can have a guide RNA customized to it. These target genes can be knocked down or knocked out via non-homologous end joining or edited with a designed nucleotide sequence via homology-directed repair. CRISPR-Cas9 has a variety of promising therapeutic applications, from chemotherapy to tissue engineering to disease modeling. Let's first focus on an example of how CRISPR can be used as an immunotherapy for cancer patients. Some cancer cells overexpress a protein called PD-L1, which binds to the T-cell receptor protein PD-1 and prevents T-cells from killing cancer cells. This resistance of cancer cells to the body's immune system is part of what makes cancer so destructive and difficult to treat. But if we can use CRISPR to knock out the PD-1 gene in a cancer patient's T-cells, this PD-1-PD-L1 pathway will be broken, and the T-cells can launch an immune response against cancer cells. Researchers at Sichuan University have been particularly active and successful in studying this application of CRISPR, and found that it is both safe and effective in a 2020 clinical trial. As is the case for genetic manipulation, CRISPR is surrounded with serious ethical debate. Using CRISPR-Cas9 to treat or cure genetic diseases appears ethically sound in principle, but scientists, policymakers, and the general public must all be aware of the slippery slope that it presents. One prominent case are the CRISPR babies, Nana and Lulu. These twins, announced in 2018, underwent illegal, unsanctioned CRISPR-Cas9 editing of the CCR5 gene as a preventative measure against the onset of HIV/AIDS inherited from their father. This was a source of great contention among the global scientific community, largely due to issues of consent and possible downstream effects or off-target edits. Human eugenics and the potential for a more pronounced disparity in health outcomes from inequitable access are major ethical concerns to be considered. Sufjan and Garland Thompson wrote, from the perspective of scholars living with genetic disabilities, we have grave worries that the use of these genetic scissors will, in the future, cut people like us out of existence. So knowing what you know now of CRISPR-Cas9, we urge you to imagine what a future with CRISPR-Cas9 would look like. How should we best balance its uses with its risks and our rights and values?